Welcome to the lecture series on vitamins and nutrition. So today, as a continuation of previous session, we are going to study about vitamin D's mechanism of action and the leftover effects concerned with vitamin D, nutrition and therapy. So we have been looking elaborately at different aspects of vitamin D. So I have been explaining you about different fat soluble vitamins, and this is the summary slide that gives you a kind of review of what are different kinds of mechanism of action that you can associate with fat soluble vitamins. So today's session I'm going to continue on vitamin D, and I'm going to elaborate a little more on the mechanism of action of vitamin D. So if you look at vitamin D, you have basically two forms of vitamin D. This we have already dealt, but I want you to look at this slide so that you would understand the significance of the structure, and then you would connect this idea into different aspects of how mechanism of action is being elicited in human beings. See, if you look at the structure, so this is a cholesterol based hormone. So if you are aware of it, I have explained about the cyclopentanoferrihydrophenanthrin ring. So this is the phenanthrin ring, this is the cyclopentano ring. So together we call it like cyclopentanoferrihydrophenanthrin ring. So this is the characteristic structure of vitamin D. So if you look at the structure, as I've told you earlier, the steroidal hormones also make use of the same structure of cholesterol. So in that way, vitamin D is basically, you could consider as a derivatized product of cholesterol. So cholesterol being the raw material, and vitamin D is basically an advanced compound which results from the metabolic product. So both from fungi and from animal source. So you find from fungi or from plant source, as you call it, the ergocalciferol and the cholecalciferol. So today I'm going to explain you a little bit more. So this slide we have already seen, but I've taken this slide again so that I can explain certain aspects of the mechanism of vitamin D, because mechanism of vitamin D is extremely essential in understanding how these category of vitamins also act like hormones in our body. So this is very, very special, especially considering vitamin D, and I would like to explain the significance of vitamin D. So if you look at the structure, vitamin D and vitamin A are the only hormones or only vitamins that have hormonal kind of property. So this is extremely unique for this category of vitamin, because most of the vitamins, as I've told you earlier, are metabolically acting as cofactors and aid in normal metabolic pathway, and we are aware of it, and we are going to see more of those vitamins, especially the water soluble vitamins. But on today's session, the focus is more on vitamin D's mechanism of action and metabolism. So you look at this, I had a different version of this slide, but I have remade this slide so that I can explain you in detail, and you can really have a feel of the structural aspect of activated vitamin D. So you look at this structure, the 7-dehydrocholesterol. So this is what is a raw material which is normally present in our skin. And sometime before I told you in the beginning of this session that when it's been irradiated, especially by ultraviolet B category of rays from the sun, because you have three categories of rays, A, B and C, and C is ultra short, and they have very good antimicrobial property but quite dangerous and can even cause skin cancer. But ultraviolet B category of rays are the ones which are responsible in converting or initiating the synthesis of vitamin D in our body, especially in the skin. So this is the raw material, and our body is capable of making 7-dehydrocholesterol from the dietary cholesterol that we take, and some of the cholesterol are also synthesized in our body in the liver. So you see over there vitamin D3. Basically, if you look into the structure, can you look at the structure? So this is the aspect where the trans orientation of the bond, and this results in vitamin D3. So you see 7-dehydrocholesterol getting converted to vitamin D3 or cholecalciferol, and here just a cleavage results in a completely activated version of the hormone. Yet it's not fully active, and that's the point what we are going to discuss today. So you have vitamin D3. Now when this vitamin goes to liver, so in the liver we have a set of enzymes. Lots of enzymes are there. Liver is quite a good organ with plenty of metabolizing enzymes over there. So the hepatocytes in the liver, they basically add, can you see over there? There is an hydroxy group added in the 25th position. So this extra hydroxy group is contributed by the liver enzymes, and this gives rise to a version of vitamin D, what we call it like 25 hydroxy vitamin D. So this 25 hydroxy vitamin D is an active form of vitamin D3. So 7-dehydrocholesterol does not have the kind of vitamin D property. Now it gets activated into cholecalciferol, or you could consider vitamin D proper, and this itself is still not active yet metabolically. So in the liver there is one more conversion. You could call it, maybe this vitamin can still bind to vitamin D receptors, but their affinity for vitamin D receptors are relatively less compared to more active metabolites. Now in the liver you have the 25 hydroxy cholecalciferol. Now this again goes to kidneys. Now in the kidney you find one more activation happens. So you see from the liver to the kidneys, and here in the kidneys, can you notice there is one more hydroxy group added over here. So what we call it like 1,2,5 or 1,25 dihydroxy cholecalciferol. So this is what the short version of this particular activated vitamin D is called, like 1,25 dihydroxy vitamin D. So this version of vitamin D is the real active form of vitamin D. So in our body, even the dietary vitamin D, even if you are going to consider taking vitamin D directly as a supplement, see this one basically happens in our body in the skin. The transformation happens in the skin, and then we get this vitamin D3, and later if you consider, this vitamin D3 undergoes series of metabolic activation in the liver and in the kidney, and finally you have this product, what we call it like the active form of vitamin D. So this is the real deal, I would say, that binds with the vitamin D receptor. So in the previous slide, so you could take vitamin D3 either as a ready-made form of vitamin D3, cholecalciferol or ergocalciferol, to undergo this dual activation, one in the liver and one in the kidney, and finally you have this product. So 1,25 dihydroxy cholecalciferol is a real active version of vitamin D. Now what happens to this vitamin D? So as I've been telling you, you do have an enzyme for it, what we call it like 25 hydroxylase. Whenever you are adding an hydroxy group to a organic compound, especially to a benzene ring, and since an addition of hydroxy group is catalyzed by an enzyme, and this enzyme is given this name because in the 25th position this hydroxy group is added, so it's called 25 hydroxylase. Okay, so here we have one more enzyme present in the kidneys. You could consider it as 1-alpha hydroxylase. Now the question is, why these two organs are given more importance, like liver and kidneys? Can't other organs or tissues in our body are capable of doing it? Yeah, first question can be answered simply by saying, yeah, these enzymes required for them are present in these organs and they are expressed in these organs. That's true. In addition to that, what we can say, kidneys play an essential role in calcium metabolism, and liver plays an essential role in cholesterol metabolism and in the storage of fat soluble vitamins and also variety of fat soluble nutrients. So it's very natural that these organs, since their role has to do with calcium and cholesterol, having these enzymes over there and the activation of this essential vitamin D into the activated form would be reasonably good if these organs could take up the role and do it, and that's what happens in physiology. So if you consider kidney and liver playing a major role in the metabolism of calcium, so together with vitamin D, there are two more hormones parathormone and calcitonin. So parathormone plays an essential role in maintaining calcium in the blood. Although we have dealt this a little bit in the earlier session, I just want to mention it right now so that we can have a connection how this all works. So parathormone released by thyroid hormone, and it's responsible for increasing the calcium level. Basically from the bone, all the calcium gets demineralized and comes into systemic circulation. And without this free calcium, muscular contraction and many of nerve conduction related functions cannot happen in our body. And now if you consider calcitonin as a negative impact of parathormone, if there is hypercalcemia, a more amount of calcium in circulation, then calcitonin is released, which will downregulate excess amount of calcium. So if there is hypocalcemia, parathormone would take up the role and help in calcium elevation in the blood. And if too much of calcium is there, let's say hypercalcemia, then calcitonin will ensure that the levels of excess calcium would be brought low by allowing calcium to go back and settle in the reservoirs of bone. So this is how homeostasis, as I've told you, the steady state way of maintaining important physiological functions concerned with calcium is handled. And now if you look at why vitamin D has to come into this loop, because without vitamin D, absorption of calcium would be completely impaired. Not only that, in this aspect, vitamin D acts like a kind of a hormone. In fact, it would be more appropriate to call it as a hormone, because the qualification for a hormone should be, it should be produced by an organ, something like there should be an origin, and then you don't have any kind of duct to carry the hormone, but blood being used as a common pathway for carrying it, and vitamin D would qualify all the functions. So the vitamin D what we consume from a dietary source needs to undergo this activation before it could be really made useful for our body. And more importantly, vitamin D has its own receptor, and that's the third aspect what we are going to discuss today, so which will be quite interesting. So now I am going to introduce you to the concept of nuclear receptors. So what are nuclear receptors? So nuclear receptors, as the name implies, are receptors present inside the nucleus. So where do you find nucleus? I would say a pictorial representation of a cell, and you see over there the outer layer is the cell membrane, and you find the nucleus inside. So there is an outer membrane and there is an inner nucleus. So you find there are receptors inside the cell. Generally we tend to consider receptors being, I would say, lock and key kind of fashion, where there is a ligand that binds to the receptor. So it's very natural that receptors would be present on the surface of cells. It's true. Receptors are present on the surfaces of cell, especially in the membrane. The membrane bound receptors are abundant, and you have the category of G protein coupled receptors. Now when we study about vitamin D, we should look at how vitamin D is able to make its presence known, or how it contributes to the metabolism of not only calcium, of variety of functions that it has to do. See, this particular receptor, see, the name for vitamin D receptor has been so designated, you call them like vitamin D receptor, okay, or VDR, we call it VDR. So this is the picture of vitamin D receptor. The structure has been already solved, and there are little more multiple PDB IDs for vitamin D receptor. So can you see over there the vitamin D receptor? It's all helical, and you do have a little bit of beta strands here, and then there is a ligand. So this ligand is supposed to be the vitamin D. So vitamin D goes and docks into this structure, vitamin D receptor. Now we are going to look at what exactly this vitamin D receptor. So before that, we should know what this vitamin D receptor family belongs to. So vitamin D receptors basically belong to nuclear receptors. That's the reason I started this session explaining you about nuclear receptors. So what are nuclear receptors? So before we understand nuclear receptors, we should also be aware that hormones elicit their function through receptors. There are a variety of hormones, as I've told you earlier. hormones come in variety of chemical structures. You do have a cholesterol based hormones, you have a peptide based hormones, you do have other variety of hormones which are derivatized amino acids, long peptides and so on. But one category of hormones, especially the cholesterol family of hormones, most of the sex hormones come under that category. Testosterone, estrogen, progesterone, corticosteroids all come under steroidal family of hormones. Now in this way, vitamin D has a very close resemblance to these steroidal hormones. So it is much easy for us to associate or relate, since these cholesterol based products of hormone basically go and bind with nuclear receptors. So it's easy for us to associate vitamin D resembling the cholesterol based structure would also naturally dock in these nuclear receptors, and there should be exclusively some receptors present over there. So these nuclear receptors are interesting receptors because their role is extremely essential. You know what they do? They basically go and ensure what kind of genes can be expressed from the DNA. So in other words, all these nuclear receptors could very well be called as transcription factors. So you see over there this definition what I have mentioned over here. Nuclear receptors have the ability to directly bind to DNA and regulate expression of adjacent genes. So it means what? Nuclear receptors are the switches that decide what genes can be expressed and what genes can be suppressed. So what these receptors do, they actually act like transcription factors. So what are transcription factors? So little bit of molecular biology when we study about vitamin D. So transcription factors mean they are those switches that decide what genes should be expressed and what genes should be suppressed in the DNA sequence. So what these nuclear receptors do, they are protein molecules. They actually act like transcription factors and go and bind to DNA sequence. Okay, so you see over there, I'll give you an overall picture of how this nuclear receptor business works. Then you can actually look into how vitamin D, being a ligand for a vitamin D receptor which belongs to class 1 nuclear receptor, regulates its mechanism of action. So in this case, I have taken the example of thyroid hormone. So if you look at thyroid hormone or any other hormone of steroidal family, they also elicit their function through nuclear receptors. So you see over there, there is an hormone which goes inside, and being a hormone which has lipophilic characteristics, it goes inside easily. Okay, and then there is this nuclear receptor. Generally, nuclear receptors are stabilized by one more protein, what we call it like heat shock proteins. So nuclear receptors and heat shock proteins together they form a complex, and here you have LBD, which is basically ligand binding domain in the receptor, and then you have the DNA binding domain. There are two different regions present in the receptor. So if you could look at the structure, can you see over there the protein, which is basically the ligand, can go and bind with the receptor. So ligand binding region is called the LBD, and here you have the DNA binding domain, which is called the DBD. So this is basically a rough view, okay, what do you call it, like the primary sequence, where you can know which region contributes to what. So all these three pictures are same, only thing the dimension in which I have represented are different. So this one is the primary sequence level, where you can have the N terminal sequence and the C terminal sequence, because nuclear receptor, in our case the vitamin D receptor, which is supposed to be a nuclear receptor, also falls under this scheme, where the N terminal, C terminal, you designate them as like A, B and F. Then you have a hinge region which connects the N terminal to the other two domains. Then you have this pink region, what is designated as D, is the DBD, DNA binding region. So this region goes and binds with the DNA. Okay, see, suppose if vitamin D is going to ensure binding of calcium and transportation of calcium, then, what you would consider as a free ligand coming up, and then you see over there the hormone basically gets the heat shock protein gets released and the heat shock protein goes away, and here you have the nuclear receptor and the hormone forming a complex. So what you see over there as LBD and DBD are basically those regions which provide binding for the hormone. So hormone being the ligand binds to this white region. If you could look at it, hormone binds over there, keeping this DBD region vacant, and this DBD region would actually be available to be bound with the DNA. Now if you look at this structure, before it could make itself available and go inside the nucleus, there is this nuclear receptor forming a dimer. So this is very interesting. The dimer could be homodimer, the dimer could be heterodimer, whatever the case. But in general, you always expect a dimer. A dimer formation is there, and this dimer formation, once it gets into this conformation, now you can say the nuclear receptor with the hormone is very active. Now as I am explaining this, I want you to be aware, this hormone, what we are explaining as being going inside, acting like a ligand and activating the transcription process all the way, you should consider this ligand, now in our case, would be vitamin D. Now it goes inside, can you see over there? There is again a pathway which tries to tell you that there is a control way of allowing the nuclear receptor dimer complex to go inside the nucleus. So you consider this. So as I have been explaining you earlier, that vitamin D and vitamin A are special vitamins because they have hormonal like function. So if you could understand and dissect how hormones would exhibit their function, you would have an insight into the mechanism by which vitamin D would also exhibit its function. Now, not all hormones have nuclear receptors, but all hormones have a receptor as a whole. So the question is, what receptor we are talking about? So there are receptors which are present on the surface of cell, and there are receptors which are present inside the cell, and there are receptors which are present in the nucleus inside the cell, and there are receptors which are there in the cytoplasm inside the cell. So there are hormones which basically interact with cytoplasmic receptors or nuclear receptors or the surface receptors. But the one which I am going to discuss today are those set of hormones which come under the category of steroidal hormones. Okay, so thyroid hormone, progesterone, estrogen, testosterone, all these category of hormones come under steroidal hormone family. And vitamin D, having a structure resembling cholesterol, has a very close association with steroidal hormones, or I would say they act very closely in tandem with many of the steroidal hormone functions. That's one of the interesting things. When people suffer from vitamin D, they sometimes, not sometimes, in most cases show symptoms resembling as if there is a steroidal hormone insufficiency. A prolonged vitamin D deficiency in a woman can sometimes lead to infertility related complications. So you see, since they are very closely associated with nuclear receptors of steroidal hormones, vitamin D deficiency is exhibited as insufficiency in steroidal hormone function. So now we are going to look at closely into the molecular mechanism by which how these hormones elicit their function. So here let's take. Now I want you to consider this steroidal hormone is an analog resembling that of vitamin D. So vitamin D or steroidal hormone both can be called as a ligand. So now this ligand, hormone or vitamin D or any analog belonging to their class comes inside the cell. Now once they come inside the cell, now you find this nuclear receptor in the cytoplasm. Nuclear receptors, before they get activated, are stabilized by a complex, what we call it like nuclear receptor HSP complex, NR-HSP. So what is this HSP? HSP is a heat shock protein. So heat shock proteins are stabilizers. So they help in preserving the protein conformation before they really get into the job. So something like, I would say, you could always consider they act like a kind of a protective head. Maybe if you look at a sword or any sharp instrument, before you really put them into use, you just put them inside the cover and keep it in a very stable manner, so that the sharp end would never become blunt. So something like nuclear receptor is being preserved or kept in a kind of active state by HSPs. But the moment hormone comes here, which is the ligand, HSP gets removed. Okay, so giving way for the ligand to go and bind. So you see there are two colors, one is a white color and one is the blue color. The white color is the ligand binding domain, which goes and binds here, and then you have the blue color, which is basically the DNA binding domain. Now before they could go inside the nucleus, the NR hormone complex or NR ligand complex gets dimerized. Okay, so when you say dimer, we mean to say you are having a doubling of the structure. It's not like the structure is getting multiplied. Two of the structures bind in a complementary fashion. Something like when they bind in a complementary function, only then their structural conformation would be available to proceed with the function what they are up to. So without this, the assembly would be incomplete. So the NR dimer is the active assembly which could now go inside the nucleus, and now once they go inside the nucleus, you see over there, they go and bind the DBD, the DNA binding domain, goes and binds over here. Where it binds? It binds to HRE. So what is HRE? Hormone response element. So basically HRE is a DNA sequence, and this DNA sequence is a signature sequence which is unique for specific hormone and hormone related functions. Now this HRE is basically the DNA binding site for the NR nuclear receptor dimer with the ligand. As they go and bind over there, immediately you have other two proteins which help them. One is a co-activator and the other one is a RNA polymerase. So you get it? So this entire thing, RNA polymerase, co-activator, nuclear receptor dimer along with the ligand now kickstart the process, and now the target genes, it means all those genes which are under the control of hormone response element start making protein. Now before they could make protein, obviously they have to make mRNA because it's a transcription process. The mRNA comes out and the mRNA gets fit into the ribosome assembly. So once they fit well into the ribosome assembly, the ribosomes make the protein, and this protein will have the function exhibited or the function demonstrated in the cell. So you see how very delicately all these components are linked. So the vitamin D what we consume basically goes inside like this, okay, and there is this unique vitamin D binding receptor which provides the binding site for vitamin D, and this entire process happens. Now the scheme what I have explained holds good for vitamin A and vitamin D. Now in the coming section we are going to look into little more details of how this ligand related mechanisms would be exhibited, and we will also look into all the pending details about vitamin D deficiency and supplementation. Thank you.